Welcome to Sigest, your fortnightly serving of digestible science from plant and food research.
Kia ora and welcome to Sigest. My name is James Sainsbury and today we're recording from Ruakura. Back in 2020, there was a bit of a shake-up in our daily lives with the emergence of an unusual coronavirus called COVID-19, and the whole world subsequently received a crash course via the evening news on the human immune system functioning and the process of vaccine development. Today we're talking to Dr. Joelle Vernest, who wears many hats here at Ruakura, including as a principal scientist in innovative plant pathology. And today Joelle is going to give us a crash course on plant immune responses and plant elicitors with many of their vaccine-like traits. Joelle, welcome.
Thank you very much. Very happy to be here today.
So before we go full immersion on plant elicitors, could you give us a bit of background on your interest in plant pathology and a feel for a couple of your favourite projects in your career thus far?
Yes. So perhaps one of the projects I'm pretty happy about is the development of biological control agent for control of a disease of apple and pear, which is called fall blight. It's actually the reason why I'm in New Zealand today. I was invited by MAVTech at the time to come and to develop a biocontrol agent, which we did, and now this product is on the market for the last 20 years. So that was very satisfying, I would say. It was also very satisfying to be part of the response to the PSA incursion on kiwi fruit. So PSA Pseudomonas syringae actinidiae, and as we know, it almost destroyed the New Zealand kiwi fruit industry. I was able to help with the recovery, and that was a very fantastic feeling to be in that position.
Amazing. And all of that leads very nicely to one of your current areas of research and the topic of today's podcast, which is plant elicitors. Before we find out exactly what a plant elicitor is, could you tell us a little bit about plant immune responses?
Absolutely. Yeah, so plants are in contact all the time with many organisms, a lot of them potentially pathogens. The plant cannot run away from the pathogen. So what the plant has done, it has developed a number of sensors that sense the presence of potential pathogen. And when a plant sense a potential pathogen, it does respond and turn on some defenses. Now, some pathogen have the ability to break through that defense and to inject into the plant cell some compounds that we call effectors. The plant has developed some sensors for that as well, and so as a second line of defense. Now, this will give rise to a localized response and can give rise to a localized resistance. But in addition to that, the plant has also developed a system where it can transfer the message from where the attack has occurred to the rest of the plant, so the rest of the plant also turn on the defense mechanisms. And there you have now a plant which is reacting to the presence of the pathogen in the environment.
And then what are the defenses that a plant is able to throw up to protect themselves from these pests and diseases?
There are a number of mechanisms. I think what perhaps is important to understand is that at the end of the day, there are fewer mechanisms of resistance than there are pathogens. So for different pathogens, the plant will have the same mechanisms of defense. Now, if you go the other way around, it means one mechanism of defense is active against a wide range of pathogen. And that is crucial to understanding the value of elicitors.
Beautiful, which leads us to the question of what are plant elicitors?
So plant elicitors basically are compounds or even sometimes microorganisms that can be used, that we use to trick the plant, believing there are some pathogen or potential pathogen in contact with the plant. So the plant believes there's a pathogen, turn on its defense reaction, and when the real pathogen comes in, it finds a plant which is already firing all its defenses, and therefore infection doesn't occur or occur at a much, much lower level.
Very, very, very clever. So where are some places that we can get these plant elicitors from?
So there are, as I said, there are a number of elicitors. if you think in terms of evolution, the sensors that the plant has developed are based on potential pathogens. So the plant can recognize different patterns of potential pathogens. So that could be sugars, that could be lipids, that could be some proteins. So we have a wide range of compounds. In addition to that, some people have developed some chemicals which do the same job and trick the plant believing there's a pathogen.
So the pest and the pathogen sheds molecules of some description, which may be protein, lipids, polysaccharides, and that's what the plant's reacting to.
Correct. Yeah. That's correct. The word shedding might not be entirely correct in the sense that some of those molecules are released because of interaction between different microorganisms or interaction between the plant and microorganisms.
Very good. So the molecules are from the pest and the pathogen, and the plant reacts to that. And then the sort of response is similar to a broad spectrum antibiotic, which then could potentially, it's not just targeted towards the pest or the pathogen that released the molecule.
Yeah, so the main difference between something like an antibiotic and an elicitor is that the antibiotic is killing a wide range of organisms. And we hope that among that wide range of organisms, there's also the pathogen, right?
Yeah.
So we're creating a bit of a vacuum by killing a lot of pathogen. And we now know that plants, just like us, do not grow in isolation. We have what's called the microbiome, which is all the organisms, microorganisms that lives with us. In actual fact, we have more bacterial cells in our body than human cells.
Yeah.
The plant by itself would not grow very well. It needs a lot of other organisms to help to get nutrients and to resist pest, pathogen, and flooding, drought, all these abiotic factors. So when you use an antibiotic or a heavy metal such as copper, you wipe out part of that microbiome. And you might create problems. You don't only solve a problem, but also create a problem. Now, with elicitors, most elicitors, not all, but most elicitors will have no antibiotic activity. They are unable to kill any microorganisms, to kill any pathogen. What they do, they trick the plant such that the plant reacts to the pathogen. So it's a very different thinking about how to go on controlling a pathogen.
Beautiful. So that's going on all the time, regardless of whether humans are there or not. What do we do as humans to leverage off this amazing dynamic? Are there examples where we apply plant elicitors to crops to help control pathogens?
Yes, we do. In actual fact, some have been used widely for many, many years. There is one that has been used on the rice for perhaps 40 years. There is another one which is very common, which is called Actigard or Bion, which is used around the world for the control of a number of different pathogens on a wide range of crops.
So where do those elicitors come from? How do humans get them, harvest them?
So not all elicitors are natural products. And actually the one I just talked about is not a natural product at all. It is a chemical which is synthesized by us. Yeah. Okay, so it is a bit better, one would say, than a chemical pesticide, but it's still a chemical. And that's the reason why that particular elicitor is not allowed for organic production. Even though the mode of action, you would think if you can trick a plant to defend itself, it's nicer than kill pathogens, than kill organisms. But no, because the origin is a chemical, it is a chemical. So we have other ways of getting elicitors, and this is one of the projects we're working right now with Waikato University, where we're looking in the natural environment for molecules that can do exactly the same job to elicit a defense reaction in plants.
That's terrific. We'll jump into the specifics of that project in a minute. But before we move off elicitors in general, you've already mentioned one of the benefits of elicitors over sort of conventional pest management, which is they tend to be a lot more targeted and they have a lot less non-targeted effects. And I guess that is some of the problems that we have with some of these conventional treatments that you might put out. What are the other benefits that elicitors can offer? And I think you also touched on one there, which is that depending on how they're manufactured, they can also be compatible with organic production.
Correct,
Yeah.
correct. Perhaps the other great benefit of elicitors is that it gives you control of a pathogen for a much longer period of time. Now, if you trick a plant to react to the presence of a potential pathogen, the plant needs a bit of time to react. So the elicitor, you would put it way before a pathogen would come in. So it's a bit like an insurance policy you take. You do not know what the weather will be in 2 weeks' time and whether in 2 weeks' time you'll have an infection period. But if you put your elicitor today, you know that in 2 weeks' time, regardless of what's going to happen, your plant has a level of protection. And so it gives people a peace of mind because they have that protection. And that protection can last for a number of weeks. So that's important.
Yeah.
In addition, if suddenly, for whatever reason, somebody would become nervous about suddenly an infection period coming in with a lot of inoculum in the environment, you can still use the normal product you've been using for many years. You know, it's not exclusive.
Not incompatible.
So it gives you peace of mind and a bit of security and a bit of guarantee without preventing you from doing whatever you would have done otherwise.
And so, as you said, it's been used commercially.
Yeah.
What is the effect on the environment, human health, quality of crop? Is there any, the plant's doing something. Is that noticeable or significant in a horticultural context?
So, yes. So, and it can go both ways. So what we do when we trick the plant believing there's a pathogen, we somehow manipulate the hormonal balance in the plant. And when you start playing with hormones, well, it can go in a number of ways. So in some cases, you might actually improve the coloring or production of your crop, but overusing it, and you might have the opposite effect. So you want to be careful on how you bring it. Now, the other thing which is, I think, important in today's context is that some of the mechanisms that lead to resistance to a pest or pathogen are similar to the mechanism that give resistance to abiotic factor. So abiotic factors are factors which are not linked to a living organisms. So what is that? It's high temperature, it is drought, it is flooding, and we know in this country we have experienced, unfortunately, quite a lot of those in recent past, and we can be sure that in the future we'll have more of those events. So being able to give an advantage to the plant or to help the plant to overcome the problem of flooding or resistance to drought would be fantastic. Yeah.
Very much so. And one last question in general about elicitors is, and it's probably might be difficult to answer, is are elicitors used in New Zealand very much? And what's the potential for our horticultural industries to benefit from the use of elicitors or new elicitors in the toolbox?
So yes, elicitors are used in New Zealand. I couldn't tell you how much it's used. One of the problems that limit the use of elicitors today in New Zealand are the cost. It is expensive. It is a molecule which is synthesized, so not available for organic growers. And if you overuse it, you might have residue problems. So because we export, we have to make sure there's no residue problems. And so, for example, for control of PSA on kiwi fruit, the elicitor, which is today on the market, can be used only 4 times during the year. Using it more or at the wrong time of the growing season, and you could end up with a crop that has level of residue that means you cannot export. So that's one of the problems.
And that leads in very nicely to the next block of stuff that I was hoping we could talk about, which is, and we've talked about elicitors generally, so I'm keen to spend the rest of our time talking about a specific project that you've got underway that you referenced just before, which is looking at green seaweed polysaccharides as plant elicitors. To start off, could you give us the elevator pitch for this project?
Yes, so the vision for that project is to be able to have a natural product coming from seaweeds grown here in New Zealand that could be used by a number of industries in the country for control of pests and pathogens. And at this stage, we're really focusing very much on PSA and kiwi fruit because it is a major problem. My background is in bacteriology, and of course, bacterial pathogen is a bit of a passion of mine. And when you think of it, is bacterial pathogen are more difficult to control, I would say, than fungal pathogen, because we have so few tools to control those bacteria. What do we have? We've got heavy metals, copper. Copper does work, but you can get copper resistance. We get accumulation of copper in the soil that can lead to phytotoxicity. We have then antibiotics, but we don't want to use antibiotics. We don't want to have a resistance in those bacteria that could be transferred to human pathogen and not having those antibiotics available to protect us. Then we have biological control agents. Some work, but they're dependent on the environment. They're dependent on the temperature, humidity. So you can't rely solely and entirely onto biological control agents, even though they have some efficacy. And so what we have as an option now is elicitors. And elicitors, I would say, have to be looked at not as the magic bullet, not as the fantastic compound that's going to solve every problem on every crop for the rest of the time. No, it is a product that has a high value and needs to be integrated into a disease management program and could really offer growers in New Zealand and even outside New Zealand a new tool to control their pathogens.
An awesome project, and it looks like you've got a really high-powered group of collaborators working on this project with you.
No, absolutely, yes. So this is a Waikato University-led project. The leaders of the project are Mary Magnuson and Chris Glassons from Waikato University, and they're based in Tauranga. But you have also other people from Victoria University, people from Te Kotai Research Institute, and it's important to have those people on board because in the vision we developed, we are growing seaweeds here in New Zealand, and so that's important. But you have also overseas collaborator, Munster, coming in. So yes, we have a very good team of people.
And you've already done some early work looking at the polysaccharides from green seaweed?
Correct. So the program we put outside and we submitted today is actually the result of a collaboration that's going on for a number of years. And as part of the work we've done today, we demonstrated that some sugar from those green seaweeds do reduce significantly disease incident on kiwi fruit after inoculation with PSA. And that works for the green-fleshed kiwi fruit, Hayward, but also for Gold 3 and the yellow-fleshed kiwi fruit. We also demonstrated that when you use the sugar from green seaweeds, we have different genes of the kiwi fruit that are overexpressed. And those genes, we targeted them because we know they are the genes that are involved in the plant defense reaction we were talking a bit earlier.
And so what does an assay look like where you test a compound like that? You extract the polysaccharides from green seaweed and then you apply it in a controlled environment, or how do you test plant elicitors?
Yeah, so that's correct. So that's exactly what we're doing, except that the extraction and purification, all that is done by the people at Waikato University who are marine biologists. So they have a special facility where they grow the seaweed. So they know exactly the genotype of that seaweed. Everything is under control. They extract with some chemists that are involved in the project. And so we receive here at Rakura a powder and we dilute that powder at different concentration. We spray it on the plant. We give the plant some time to react, to recognize that product, to turn on its defense reaction, and then we inoculate with PSA. High concentration of PSA. We then have conditions that are highly favorable for the development of the disease. And 3 weeks later, we compare how much disease we have on the treated plant versus the non-treated plants. And we can compare different timing, we can compare different concentration of the same sugar, and we can also compare different sugar from different seaweeds. And that is important. That's part of that program with the Waikato University we're talking a bit earlier, where we know that sugars from some seaweeds are very effective. Some are more effective than others. Yet they're very similar. And chemists can tell us here is a small difference. And the question now is this small difference the reason why this polysaccharide or this sugar is more active? And finding that is what we call structure-function relationship. Finding that would be fantastic because now we know exactly what sugar we're after to have a consistent high level of protection of our crops.
Yeah, terrific. So when the polysaccharide's extracted at the moment from the seaweed, it's a mixture of sugars and molecules, or it's a pure sort of...
It is at a level of purity.
Yeah.
It is a level of purity. So it's not a bag of green seaweed that is mixed. And no. It's much more than that. There is much more than that. There is different steps. So at the end, we know that what we have is the sugar.
Yeah.
The sugar we're talking about are long chain of molecules. So during the process, they can break down. So the exact size, is a mixture of sizes of the same polysaccharide.
Because that observation that you made about sort of we're talking down at the molecular level, so subtle changes in the molecule can have a huge impact on the functionality, and as well as presumably the plant or the source of it, can external environmental factors affect the sugars that a plant may produce, even if it's genetically the same, could produce a different type of sugar. And then I guess part two of that, are humans able to manipulate molecular structures, or is that in a systematic way and test?
Yes. So there are a number of questions in that one question. So number one is, yes, algae or seaweed that are genetically related will produce sugar which are slightly different. And that small difference results in huge differences in plant defense. Those sugar can be modified, and chemists know how to modify, remove a sulfate group here or add one, or they know how to do that. But there are people involved in this project from Victoria University who have discovered some microorganisms living with the seaweed, and those microorganisms produce naturally some enzyme which naturally modify the sugar. So as part of the vision is really harvesting some seaweeds growing here in New Zealand, extracting the sugar, modifying it with enzyme produced by organisms coming from the environment to a point where we have a product we know will give a high level of control.
Wow.
Yes.
and then the high throughput assay allows you to really screen all of these things in a very, very methodical way. So amazing.
Yeah. There's some screening that needs to be done. However, we're already well on the way in the sense that we do have several sugars that we know control PSA on kiwi fruit, green-fleshed, yellow-fleshed kiwi fruit. They also control other pathogens on tomato, for example. And the chemists at Waikato University, they know the structure. They know where the sulfate group is, where the other group are, and yeah. So we're very well on the way already. So yes, it looks like a vision for the future, but it is an achievable future. It is a vision that I truly believe we can achieve in the medium term. Yeah.
Yeah, very, very, very clever and very exciting. So as we move towards the end of the interview, I'm keen to widen the lens a little bit and ask you to speculate about, in 10 years' time, do you think elicitors will be much more widespread in terms of their usage in New Zealand? They'll be more available. It will be more of a conventional and affordable disease management tool in plants?
I do believe absolutely, and a number of reasons for that. If we look outside in New Zealand, if you look, for example, in Europe, there's a very strong push to get rid of many chemistries that are used today for control of pathogen. And that we're talking now fungal and bacterial pathogens. So there is a need to replace those chemistry with new products. Because population is growing, we need to produce food. So the need to produce food is still there. And so we need to protect the plants from the pathogen. So elicitors represent a fantastic opportunity to fill the gap that's going to be left by all those chemistry that won't be allowed in the near future. And as I said, there's also the case of the bacterial pathogen, which are much more difficult to control. And now that we're moving toward natural product as elicitor, because in the past, as I said, we had some chemicals as elicitor. So now with moving to natural product, I think it will be even more acceptable for people in New Zealand and outside New Zealand to use them to protect their plants.
So the polysaccharides from green seaweed sound very, very promising, but I've got to ask you, why seaweed? Why is that the focus of this particular project, and sort of are there other potential areas as well?
Yeah. So seaweeds are part of a marine environment, and the marine environment hasn't been really explored. So there are a lot of organisms growing there, and algae and seaweed are one of them, that produce compounds which might have fantastic capabilities or could offer us something that hasn't been explored so far. So, yeah, we believe that we're going to find in the marine environment quite a lot of compounds of great interest. I believe, in actual fact, that some anti-cancer molecules have been found from algae growing actually in New Zealand. And the people in Waikato University have been working in that space for many years. And so we have here in New Zealand some international experts in that field.
Yeah, beautiful. So leveraging off the untapped potential of the marine environment and amazing expertise that's on hand.
Absolutely. Yeah,
excellent.
That's correct. Yeah.
And so I think that is a great place to wrap up. Joelle, you've taken us on a journey into the world of plant immune responses, and through your exciting research focus, shown us the potential of elicitors to change the face of modern horticulture. Thank you.
Thank you. Great pleasure to be here today.
And thank you out there for listening. Please check out the Sigest pages on our Plant and Food website for extra links and to see our full catalogue of podcasts. Please follow or subscribe to our channel on iTunes or your favourite podcast provider. And to help this channel grow, please post a review and rate this podcast. Mā te wā, until next time.